Great. Thanks very much, Grazi. It's wonderful to be here, even at such an awful hour of the morning, which it is for me. David's going to be talking about our experimental data. When I say our experimental data, of course, I really mean his experimental data. And I'll be talking about the models. So, since you always have to start with the data before you do a model, David is going to take over the first part of the talk. And so, David, over to you.
Okay. So, all the data that we're going to present is actually from exocrine glands. And so what the models have been trying to interrogate is stimulus secretion coupling in exocrine glands. So these glands are, you can think of them like a bunch of grapes, essentially where one cell type, the acinus, is the grape, and the other cell type, the ducts, is the stalk. And the cells take, they take stimulus onto the acinus, and they secrete from the acinus into a ductal system, which ends up secreting fluid and enzymes into your mouth or into your gut. So next slide. Over the last 25 years or so, James and I's collaboration has been to model fluid secretion in the salivary gland. And essentially, without going over all the details, what the gland is trying to do is move chloride from the blood into the ductal system, and that will allow sodium to follow and water to follow the sodium chloride, giving you a saliva which is sodium chloride rich. So next slide. The impetus for this secretion is obviously the sight, the smell, and the thought of food. So when those things occur, next slide, there is a nervous input through the parasympathetic nervous system, which releases acetylcholine, next slide, and that causes an increase in calcium within the acinus cell, which actually activates that chloride channel and gets the whole system working. So next slide. So we have gathered a bunch of information that James has used in the modeling over the years, and this has been in the form of gathering spatial information, through calcium imaging experiments, and temporal information by looking at the calcium signal actually in cells. And the principles that we've established that the model has been built on is that there's both spatial and temporal information, and the fundamental facts are that we get long, slow calcium oscillations when we stimulate with the neurotransmitter. So in the right panel, you see cells being stimulated with carbachol, which is an acetylcholine analog. And we essentially get long, slow calcium oscillations. And the other fundamental characteristic we can see from the movies are that we get global calcium signals. So the signals have spatial information. So next slide. To illustrate these global calcium signals, that these signals are invariably global, what we actually do is we produce the stimulus for the calcium signal in one end of the cell. And so that's essentially releasing IP3 using a laser in a small volume in the blood face of the cell. And if James plays the movie again, you'll basically see that even though we produced the calcium signal, we initiated the signal in the basolateral phase, the majority of the signal initiates in the apical phase, but invariably becomes global. So these factors were actually the fundamental basis of the model that James has developed over the last 5 or so years. Next slide.
All right, so that's where I come in. That's the basic experimental information. And the question is, how do we write down a model of all this stuff? Actually, the first question one should always ask, of course, is why should we model it? But our modeling goal is to understand what on earth is going on. So let me give you a very brief primer on how to model calcium in cells. I'm sure most of you know this already, but for those who don't, here's a very brief introduction. The cytoplasm of a cell has a very low calcium concentration, extremely low. That's because calcium is in fact, or at least free calcium ions are in fact toxic. They turn stuff on and they turn stuff off. And if the calcium goes up too high for too long, the cell will die. And so the cell spends a lot of energy keeping its cytoplasmic calcium concentration very low. On the other hand, the calcium outside the cell is extremely high. This is about 4 or 5 orders of magnitude bigger than the calcium in the cytoplasm. Not to mention that the endoplasmic reticulum also has a very high calcium concentration, as does, well, the mitochondria is not all that high, to be honest. So that means that the cytoplasm is under intense calcium pressure. And all you have to do is open up these calcium channels, open up a leak here, say, or open up an IP3 channel, IP3 receptor, which is a calcium channel, or a ryanodine receptor, which is also a calcium channel. You open up those channels just a little bit and calcium rushes in, which means that the cytoplasmic concentration goes right the way up. Very fast. Well, you know, not fast compared to neurons, for example, but fast compared to, I don't know, the speed at which I run, for example. Then in order to get that calcium concentration back down, you just have to shut these channels. So these channels go shut, and then circuit pumps, sarcoplasmic and endoplasmic reticular calcium ATPases, take the calcium out of the cytoplasm and put it back into the ER, or they take it outside of the cell. And by this sequential opening of calcium channels and then reuptake by the calcium ATPase pumps, you get the calcium concentration in the cell going up and then down and up and then down. And it is dogma that the signal is carried by the frequency of that oscillation of calcium. So, you know, a fast one is sends the regulation network a slightly different signal than does a slow one. But we shall talk more about that in a minute. I say it's dogma, but does not actually appear to be true for our cells here. You construct the model, you take each of these various bits and pieces, you know, the pumps and the calcium fluxes in and the ryanodine receptor and the IP3 receptor, and each of those has its own somewhat complicated model, which people have worked on over the years, and you decide which bits to put in, and you grab those models and you stick them all together, you connect them up, and that gives you your overall model. So for example, for our cells, for parotid Acinar cells, we know that you have to have circuit pumps and plasma membrane pumps. You've got to have store-operated calcium channels, and you've got to have an IP3 receptor. You also have to have buffers, but I didn't put that arrow in. I'm not sure why I didn't, but I didn't. And most of these other things are probably not as important for the kinds of questions we're asking here. So you build the model in a modular kind of approach, so it can be adapted to many different other cells as and when needed. That's our calcium toolbox. So how do these cells really work? I talked about the calcium so far, but there's all the other ions, the chloride, the sodium, and the potassium ions, not to mention the bicarbonate and protons, which are really, really important. As David said, it is the transport of chloride ions from the outside of the cell, from the interstitium into the cell, and thence into the lumen. It's that transport of chloride followed by the flux of sodium that concentrates sodium chloride in the lumen, and that draws water from the interstitium through the cell into the lumen by osmosis. And therefore cells have got to have very well regulated ion transporters to make sure they keep their volume stable and to make sure that they can transport chloride as and when required. So in the basal membrane of the cell, which is the one facing the interstitium, you have a sodium-potassium chloride transporter exchanger, you have a sodium-proton exchanger, you have an anion exchanger, all these bits and pieces. You have a sodium-potassium ATPase, you have a calcium-activated potassium channel, etc., etc. And in the basal membrane of the cell, you have calcium-activated chloride channels. So you have rather different sets of channels in the apical membrane than you do in the basal membrane. And of course, you have bicarbonate buffering and pH control in the middle of the cell. For the calcium, you have the IP3 receptors sitting close to the apical membrane of the cell. We know that's where they sit because David did a wonderful experiment where he labeled them with his high-performance, super-duper, extra special plus highly expensive microscope, and all the IP3 receptors, you could see the little green blobs sitting really, really, really close to the little red blobs, which were these chloride channels. So most of what I say is, in fact, well justified by detailed experimental evidence, although I'm quite sure not all of it is justified by evidence, as is my way. Okay, and so when you get calcium released from the IP3 receptors, which are sitting in this part of the cell, that creates the back propagation of a calcium wave back through the cell in order to stimulate PLC, phospholipase C, which makes IP3 in the basal membrane of the cell. Therefore, according to our thinking, the back propagation of this calcium wave is vital for how these cells secrete water. So here's we'll just briefly go through that. Here's the basal region. Here's the apical region. When you bind agonist, it makes IP3 in the basal region. That makes IP3, diffuses through the cell. It then binds the IP3 receptors in the apical region of the cell. Those IP3 receptors release calcium. So calcium is released in the apical region. That stimulates a back-propagated calcium wave, which goes back to the basal end of the cell, and that lets us stimulate PLC and make an oscillation of calcium. So that was our current thinking, and in fact, that's sort of the dogma in the field, and it's been that way for many years. What else did we do? Well, we actually built this into a full three-dimensional reconstruction. Again, David used a super duper fancy, expensive microscope, which has a special name, which I can't remember, and took optical slices through a group of 7 cells. And we reconstructed those optical slices into a three-dimensional finite element reconstruction of those 7 cells, which is this group here, and together with the associated lumen. So this is the lumen that collects the water. These are the cells which are clustered around this bit of lumen. Here's the original data on the left. And what you can see is that each cell is sort of surrounded by the lumen. There are fingers of it. It's like the cell is a fist and the lumen is a bunch of fingers which sort of go around it like this. And this is the case for every cell. And each part of this lumen is shared between, in general, 2 cells. We then solved our model equations on that group of cells, and we tried to understand how our mechanism was working to secrete saliva. These are typical results. We could show a lot more, of course. Here's a lovely movie by John Rugas, who has done lots of the programming for what you see here. I didn't write the programs, of course. I get paid far too much to write programs, so we hire someone else to write programs. Many years ago, it was Krasi's turn to write programs for me. Thank you, Krasi. But now it's somebody else's. Here are typical responses from the calcium oscillations from the apical region, from the basal region, shown here. Here's the rate of fluid flow from the cluster. As you can see, you get this weird sort of MMO here. That's only because you have 7 cells, each of which is oscillating at a slightly different frequency, and so you get this summation of the fluid coming out of each cell, giving you this mixed mode pattern here. And here are the concentrations in the cell and the lumen of carbon dioxide, chloride, bicarbonate, potassium, etc. So all these concentrations check out very well. The calcium oscillations check out very well. The fluid flow checks out very well. And we concluded that, well, we kind of understand what's going on, even in the three-dimensional thing. I mean, there are questions to do with the spatial distribution of the calcium. There are still questions, but we figured we kind of understood it. That was all very well, but then came Jonathan Rubin's spanner in the works, where I head back over to David.
So, you know, the dirty little secret about all the data that we produced over the last 20 years or so, up until this point, is it's been produced in isolated cells that we take from a gland and we digest by enzymes, and therefore it's not in its native environment. I mean, that's for sure. I mean, it has no interaction with extracellular matrix or basement membrane. And another issue with our previous data is how the agonist is actually presented to the cells. We essentially just add a square wave pulse of agonist and we monitor the response. And that's clearly not what is happening when the gland is stimulated by a nerve. So we decided that we would try and measure some of these parameters in vivo using intra-vital microscopy. So essentially we take an animal which is expressing a genetically encoded calcium indicator, and we use multiphoton microscopy to interrogate the fluorescence from this dye in a living mouse. So next slide.
I'd forgotten I was doing the slides.
Yeah, you can play the movie as well, James. So essentially we breed a mouse that will drive the expression of the dye of the probe in one particular cell type. So this is in the Acinar cells. So GCAMP6F is the indicator, the MIST is the promoter which drives this expression in Acinar cells. So you can see these big black holes in the middle here. These are essentially where the ducts would be. So all we're measuring from is Acinar cells. So next slide. So we can monitor the calcium signal in slabs of tissue in vivo, and we can stimulate a nerve bundle that goes into the gland with different frequencies of stimulation. Here we're stimulating with 5 Hertz stimulation to the nerve and monitoring the calcium signal in regions of interest that you can see that James is pointing to. And essentially what we see here is, again, we're seeing calcium oscillations. We're recording at 10 frames a second. And the first difference that we see is that we still get calcium oscillations, but they're actually much faster than we were seeing in isolated cells. So next slide. The next thing that is different from experiments that we did previously is if we zoom in a little bit now and stimulate with a lower frequency. Now, in contrast to the isolated cell experiment, where we invariably saw global responses, here we're seeing highly localized responses around the apical end of the cell. You can see this with these flashes of yellow and red around the lumen. So we're getting apically limited responses, again, which are occurring quite rapidly. Again, this is recording 10 frames a second. So next slide. So we can visualize where the response is occurring by taking that whole movie and generating a standard deviation plot of the whole movie sequence of the 600 frames. And you can see if we zoom in on one particular acinus, which is probably about 10 cells, and you look at this volcano plot, again, it's pretty obvious that the signal is in the apical end of the cell and is really not globalizing to the basolateral regions of the cell. So next slide. If we measure the calcium signal again in that Acinar cell and essentially just look at the fluorescence within regions which represent the apical or the basal region, you can see that you get these much faster calcium oscillations which are restricted to the apical region of the cell. They're not globalized. There's pretty much nothing going on in the basal regions of the cell. So that's another difference from the in vitro data. So next slide. We also noticed that there was heterogeneity across the field that we were looking at. So if we stimulated the glands at 3 Hertz, we'd have a portion of the cells that increased their calcium. If we increased the frequency of stimulation, we would recruit more of the field of view to respond, but the initial responding regions appear to always respond first. And so to analyze this, we decided we'd take an approach to divide the field of view into a grid. And you can see that here. We divided it into an 8x8 grid, 32 microns by 32 microns, and then essentially process the image as a standard deviation image within each of the 32 grids. So next slide. So what we did here to look at the heterogeneity across the responding grids was to essentially rank the grids based on the magnitude of the response in a particular grid. So the the grid that responds greatest here is G6. It has the lightest gray. And this is analyzed from a frequency where all the cells respond. So next slide. If we now plot the same calcium response based on that same ranking for low frequency and high frequency, you can see that generally an area of the field of view responds first and biggest to all frequencies. So we're interested in why that happens. So next slide. And we rationalize that this could be that there's actually a heterogeneity of neural distribution within the field of view. That's one thing that could explain this. And so a particular area of the field of view is preferentially stimulated. The other explanation could be that there's a homogeneous nerve distribution, but as we increase the frequency of stimulation, we recruit more of those nerves to actually fire, more of those neurons to actually fire. Another alternative, I guess, is that the Acinar cells could have a heterogeneous distribution of neurotransmitter receptors on the cells, such that the ones with the most receptors respond first. So we're interested in sorting this out. I'm going to tell you we don't have the answer, but next slide. To sort of at least address the first issue, that there's a heterogeneity of neural inputs, we did was actually cross a mouse that expresses membrane-bound red fluorescent protein in all cells with a driver that would put a green fluorescent protein in cells that express acetylcholine transferase, i.e. they're representative of parasympathetic neurons. And you can see that there's a relatively homogeneous distribution of neurons through the field of view. So we don't think it's that there's a heterogeneous distribution of neurons. And to get at some of the other issues, next slide, we've crossed that same, so Jane.
Was that the right one?
That's the right one. I just was worried that you hadn't had enough tea this morning. So we've crossed this GCamp mouse with the cholinesterase transferase mouse. So now we're actually monitoring the calcium signals in the parasympathetic neurons, and we can monitor those signals. And our first indication is that that at higher frequencies we get more cells to respond, but this is a work in progress. So presented with that new data, it's over to James to sort this out as far as the modeling goes.
Right, so there's been a lot of developments just in the last, I don't know, 2 or 3 months or something. It's very recent. None of this is published yet, I don't think. Is it, David? We haven't done anything yet? No. And the problem is that David's new data have shown us quite clearly that our previous model simply doesn't work. It's just wrong. Our previous model, you might or might not remember, is that you had a slow calcium oscillation, and you had to have a back propagation of a calcium wave. Without the back propagation of a calcium wave, it doesn't work. The cells don't secrete saliva. And yet when you look at it in real animals, there is no back propagated calcium wave. There is an isolated cells, but not in real animals. And therefore we actually have to construct a completely new model, which goes against the current dogma of how these cells work. And we haven't done this yet. So we actually have no answer. I mean, but this is what we think is going to happen. There is, I should add parenthetically, quite a lot of data that most of the channels and things and transporters that are in the basal membrane also sit in the apical membrane, possibly at lower density, but they seem to be there. So for example, we've shown that there's an ATPase in the apical membrane, which was unexpected. We've shown there's a calcium activated potassium channel in the apical membrane, which again was unexpected. But that means that all our previous data over the past 5 years, which was previously somewhat of a puzzle, we didn't understand why those things were in the apical membrane. We now believe we do understand why they are. And it is because the apical membrane of the cell seems to be the only part of the cell which sees a rise in calcium concentration. And therefore, anything to do with fluid secretion, all those channels, they have to sit in the apical membrane of the cell, or the cell doesn't work. And that's going to be our new hypothesis, is that in fact a parotid acinar cell, although it's a polarized cell, actually does all the work, or most of the work anyway, in the apical region, which contains all the necessary transporters and calcium bits. Will it work? We don't know. Will it transport water? We don't know. Will it be able to maintain a stable cell volume, which is quite difficult to get in a model? Again, we don't know. But I suppose time will tell. So David and I will keep our fingers crossed while we go and construct this new paradigm and submit it for publication, I would guess. And I think that is probably time, in which case we are done. Just a list of collaborators. I just do want to give a special shout out to John Rugas, who is a scientific programmer in Auckland and did a lot of the pictures that you saw today. And I think that's it from us.
Thank you very much, James. Really interesting. Maybe I can start with asking a question about your previous picture, the new view of the cell. So you have here calcium sensitive potassium channels. Why are they there?
Okay, so the reason you have a calcium activated potassium channel is because you have a calcium activated chloride channel and you want to transport chloride. So when the calcium goes up, it activates a chloride channel, which lets chloride flow through, but that would depolarize the membrane. And so if you were to not compensate for that, you would have the membrane depolarize, and that would stop calcium flux, which you don't want. So you have to re-hyperpolarize the membrane, or keep it hyperpolarized, in order to maintain calcium flow. And that's what this potassium channel does. When calcium activates the potassium channel, it then pulls the membrane potential back down and keeps the membrane potential high and negative, allowing for continued chloride flow. The dogma says that all those channels, all those calcium activated potassium channels, sit in the basal membrane of the cell. That doesn't work. The model shows quite clearly you cannot have that and have it consistent with the data from real mice. Therefore, those channels have to sit in the apical membrane of the cell. We have in fact shown experimentally that they are there previously.
But you will then have you have a response actually in the membrane potential when the cell is going to be kind of excitable. I mean,
yeah. Well, it's not actually an excitable cell, but your apical membrane potential and your basal membrane potential, they are both different, and they're both going up and down, up and down, up and down, up and down. Yeah, for sure.
Yeah, we have a question from Victor, but just before that, I want to ask one question, sorry, Victor, to David. David, when you stimulate with this 3 hertz, 2 hertz, 1 hertz, 5 hertz, is this optogenetic stimulation?
No, essentially there's a nerve bundle that goes into the gland, and it's just wrapped with a platinum electrode.
So you can access it from the outside?
Yeah, we can access it from the outside. Yeah, no, optogenetic stimulation would be very elegant, and that's an interesting idea.
Actually, so, I would like to jump in and point out that David tried to do an experiment where he just made the mouse smell cheese and tried to get the oscillation.
I thought that would be so cool that if it smelt and salivated and we could see the calcium signals, that would be brilliant. And we tried. We tried cheese, that didn't work. We tried Japanese unami bomb, that didn't work. And so then we rationalized the fact that mice actually, that we actually didn't make them smell it. We tried to get them to taste it. And so I think that's the issue. Maybe mice don't taste, they actually do smell. So we haven't done the smell experiment, and that's on the plate.
Yes, sure. Victor says peanut butter works best. But seriously, the question, he's asking how compartmentalized is the apical and distal parts. So how, I can't pronounce it very well, but how separate.
Compartmentalized, yeah.
Yeah. Is there very special, is there some very special cytoplasmic organization?
In our model, no, Victor, there isn't. It's a really good question. The compartmentalization is not built in directly. It's simply a result of the low diffusion coefficient of calcium. So you have a single domain where you have calcium diffusing with a very low diffusion coefficient, around about 5 to 10 microns per second. But the other ions, the chloride, potassium, and sodium ions, they all have a much higher diffusion coefficient. So effectively, the chloride, sodium, and potassium are homogeneous in the cell because they diffuse so fast. IP3 is, as we discovered, effectively homogeneous in the cell because, again, IP3 diffuses fast enough for that. But the calcium is not homogeneous in the cell because of its low diffusion coefficient. And that's all you need to have the difference between the apical and the basal regions. You, of course, have to have a resistance between the apical membrane potential and the basal membrane potential. And that resistance is